Hi everyone, and welcome to my channel. Today we're going to talk about cells, the basic unit of life. All living things are made up of cells, either single cells, or many cells. Cells are the smallest units that can carry out all the functions of life, such as growth, reproduction, and metabolism. There are two main types of cells: prokaryotic cells and eukaryotic cells. Prokaryotic cells are the simplest type of cells and are found in bacteria and archaea. Eukaryotic cells are more complex and are found in plants, animals, fungi, and protists. The basic structure of a cell is the same for all types of cells. The cell is surrounded by a membrane that protects the cell's contents. Inside the cell is the cytoplasm, which is a jelly-like substance that contains the cell's organelles. Organelles are specialized structures that carry out specific functions in the cell. Some of the important organelles include nucleus, mitochondria, endoplasmic reticulum, Golgi Apparatus, lysosomes, vacuoles, etc. Let's discuss them in detail. Let's kick things off with a brief introduction. The cell membrane, also known as the plasma membrane, is like the gatekeeper of the cell. It's a phospholipid bilayer that encases the cell, controlling what enters and exits. But there's so much more to discover. Now, let's delve into the cell membrane's structure. Imagine a sea of lipids, with proteins and carbohydrates floating within. The lipid bilayer comprises two layers of phospholipids, each with a hydrophilic head and hydrophobic tails. Proteins are embedded in this lipid sea, some spanning the entire membrane while others rest on the surface. Carbohydrates decorate the outer surface, forming the glycocalyx. One of the membrane's key functions is selective permeability. This means it allows certain molecules to pass through while keeping others out. Small nonpolar molecules, like oxygen and carbon dioxide, can slip through the lipid bilayer. But larger molecules, ions, and polar molecules need help from specialized transport proteins. Let's dive into the various ways molecules move across the membrane. Active transport uses energy to move molecules against their concentration gradient, while passive transport relies on the gradient and doesn't require energy. Diffusion, facilitated diffusion, osmosis, these processes keep the cell in balance with its environment. The cell membrane is like a communication hub. Receptor proteins on the surface recognize specific molecules, like hormones, and trigger cellular responses. This is how cells communicate with each other and their environment, ensuring proper coordination. Did you know the cell membrane also plays a role in cell adhesion and forming tissues? Proteins like integrins help cells stick together and interact with the extracellular matrix, providing structural support and influencing cell behavior. When substances are too big to pass through the membrane, cells have ingenious methods. Endocytosis engulfs particles by forming vesicles, while exocytosis releases materials outside the cell. These processes are essential for nutrient intake, waste removal, and cell communication. All eukaryotic cells have something in common: they contain a nucleus, which orchestrates numerous cellular processes. Let's take a closer look at the structure of the nucleus. The nucleus is usually located near the center of the cell and is surrounded by a double membrane envelope called the nuclear envelope. This envelope has tiny pores that allow for the exchange of materials between the nucleus and the rest of the cell. Inside the nucleus, we find the nucleoplasm, a gel-like substance that houses various components. One of the most crucial components is the nucleolus, responsible for producing ribosomes. These ribosomes play a key role in protein synthesis. Surrounding the nucleolus, we have chromatin, which is a complex of DNA, RNA, and proteins. Within the chromatin, we have the DNA, the genetic blueprint of life. During cell division, the chromatin condenses into distinct structures called chromosomes. These chromosomes contain genes, which carry the instructions for making proteins and governing various cellular functions. Now, let's discuss the vital functions of the nucleus. The nucleus is the keeper of our genetic information. It controls the cell's activities by regulating gene expression. When a cell needs to make a specific protein, the relevant part of the DNA is transcribed into RNA, which then travels to the cytoplasm for protein synthesis. Another critical function of the nucleus is its role in cellular reproduction. During cell division, the nucleus orchestrates the replication and distribution of genetic material to ensure that both daughter cells receive the correct amount of genetic information. The nucleus is also involved in cellular communication. It receives signals from the cell's environment and responds by adjusting gene expression. This enables the cell to adapt to changing conditions and maintain its balance. In summary, the nucleus is like the brain of the cell, governing its activities through the control of gene expression, facilitating cell division, and participating in cellular communication. Before we jump into the nitty-gritty of the endoplasmic reticulum, let's lay down the basics. Imagine a cell as a bustling city, and within that city lies a bustling factory, the endoplasmic reticulum. Here's what the ER looks like. It's not just a static structure. It's a dynamic network of membranes intricately woven throughout the cell. Picture it as the cell's very own highway system, connecting various regions and enabling the efficient movement of materials. Now, imagine this network split into two main types, the smooth ER and the rough ER. The rough ER stands out due to its studded appearance, thanks to ribosomes dotting its surface like busy workers in a factory. These ribosomes are the cell's protein-producing powerhouses. As the ribosomes work tirelessly, they synthesize proteins that will serve diverse functions within the cell and even beyond. These proteins can range from enzymes that facilitate chemical reactions to structural proteins that give the cell its shape and integrity. Producing proteins is just the beginning. Endoplasmic reticulum takes its quality control seriously. It ensures that the proteins are properly folded, and if any fail the test, they're sent for recycling. On the flip side, we have the smooth ER, which lacks these ribosomes. But don't let its smooth appearance fool you. It's a hub of activity too. Smooth ER is like a lipid factory. It's responsible for synthesizing lipids, which are essential components of cell membranes, hormones, and other crucial molecules. These lipids are then dispatched to different parts of the cell, contributing to its structure and signaling pathways. Hold on to your seats, because endoplasmic reticulum also plays a pivotal role in intracellular transport. It's like a busy highway network within the cell, allowing materials to move from one part to another in tiny bubble-like structures called vesicles. But wait, there's more. The smooth endoplasmic reticulum isn't just about lipids. It's also responsible for detoxifying the cell. It's like the cell's detox center, breaking down harmful substances. Get ready to uncover the secrets behind ribosome structure, their strategic locations, and the incredible impact they have on life itself. Before we dive into the nitty-gritty, let's start by understanding the structure of ribosomes. Ribosomes are composed of two subunits, the larger subunit and the smaller subunit. These subunits come together during protein synthesis, but can also exist independently in the cell. The larger subunit contains three binding sites: the A site or aminoacyl tRNA site, the P site or peptidyl tRNA site, and the E site or exit site. The smaller subunit plays a vital role in positioning the messenger RNA during translation. Ribosomes are found in various locations within the cell, reflecting the dynamic nature of cellular processes. Let's explore these locations. The majority of ribosomes are located freely in the cytoplasm. These ribosomes synthesize proteins that are intended to stay within the cell itself. Some ribosomes attach to the rough endoplasmic reticulum, creating the rough appearance due to ribosomes studding the ER's surface. These ribosomes synthesize proteins that are either secreted from the cell or are embedded within cellular membranes. Mitochondria, the powerhouse of the cell, have their own ribosomes. These ribosomes are responsible for producing proteins that are essential for mitochondrial function. Now that we've grasped the structure and locations, let's discuss the immense importance of ribosomes in the cellular world. Ribosomes are the workhorses of protein synthesis. They read the genetic information encoded in mRNA and translate it into functional proteins that carry out numerous biological functions. As cells grow and divide, they require a constant supply of proteins. Ribosomes play a pivotal role in ensuring that cells have the necessary proteins to maintain their structure and function. Ribosomes don't just produce proteins for structural purposes. They also synthesize enzymes, signaling molecules, and regulatory proteins that orchestrate cellular activities. Let's now delve deeper into the functional aspect of ribosomes. The ribosome initiates protein synthesis by binding to the mRNA and assembling the two subunits around it. This process involves initiation factors and a special tRNA called the initiator tRNA. During this phase, the ribosome moves along the mRNA, reading the codons and recruiting appropriate tRNA molecules that bring in the corresponding amino acids. Peptide bonds are formed between these amino acids, creating the growing polypeptide chain. The ribosome recognizes a stop codon on the mRNA, and this signals the end of protein synthesis. Release factors help in the disassembly of the ribosome and the release of the newly synthesized protein. Mitochondria are tiny, double-membrane organelles found in most eukaryotic cells. That's cells with a nucleus, like ours. These powerhouses are often referred to as cellular power plants due to their role in generating energy. Now, how do these mighty mitochondria produce energy? Well, they perform a process called cellular respiration. This involves breaking down nutrients, like glucose, through a series of chemical reactions to produce ATP, the energy currency of the cell. Let's take a closer look at the structure. Mitochondria have an outer membrane and an inner membrane, which are highly folded into structures called cristae. These folds increase the surface area for reactions, making energy production even more efficient. Let's delve into the intricacies of mitochondrial membrane structure and transport systems. The outer membrane acts as a protective barrier, while the inner membrane houses the machinery for energy production. Transport proteins embedded in these membranes shuttle molecules and ions, playing a vital role in maintaining the electrochemical gradients necessary for ATP synthesis. Did you know that mitochondria are quite the shape-shifters? They constantly undergo processes of fusion and fission. These dynamic changes influence mitochondrial function, distribution, and overall health. Think of it as a cellular ballet where balance is key for optimal energy output. Now, here's where things get mind-blowing. Mitochondria have their own DNA, separate from the cell's nucleus. This has led scientists to propose the endosymbiotic theory, suggesting that mitochondria are descendants of ancient bacteria that were engulfed by early eukaryotic cells in a mutually beneficial partnership. Let's talk evolution. The relationship between early eukaryotic cells and mitochondria was a game-changer in the history of life. This symbiotic partnership provided the energy boost needed for complex cellular functions and eventually paved the way for the diversity of life forms we see today. But wait, there's more. Mitochondria aren't just energy generators. They're also involved in various other essential functions, like regulating cell death, calcium storage, and even influencing cellular signaling pathways. Did you know that mitochondria also play a role in cellular communication? They release signaling molecules that influence various cellular processes, including metabolism, growth, and even immune responses. It's like they're not just power plants, but also cell phone towers. Now, let's discuss the link between mitochondria and health. Dysfunction in mitochondria can lead to various diseases, collectively known as mitochondrial diseases. These can affect the nervous system, muscles, and even the heart. Interestingly, mitochondrial function is also linked to the aging process. As we age, the efficiency of our mitochondria can decline, impacting overall energy production and potentially contributing to age-related diseases. But fear not. There are ways to support and maintain healthy mitochondria. A balanced diet rich in antioxidants, regular exercise, and reducing stress can all contribute to optimal mitochondrial function. Researchers are continually uncovering new insights into mitochondria. Recent discoveries have shed light on mitochondrial dynamics, interactions with other cellular structures, and their role in diseases beyond just energy-related ones. It's a thrilling time for mitochondrial research. Looking ahead, mitochondria are even stepping into the realm of biotechnology. Scientists are exploring ways to harness their unique properties for applications like targeted drug delivery, bioenergy production, and even potential therapies for mitochondrial diseases. The future is indeed bright for these cellular powerhouses. Prepare to be amazed as we uncover the story behind its discovery, unravel its complex structure, and shed light on the pivotal role it plays within the cell. Without further ado, let's embark on this illuminating adventure. The story of the Golgi apparatus begins in the late 19th century, when an Italian biologist named Camillo Golgi made a groundbreaking discovery. In 1898, Golgi used a staining technique to visualize what he termed the internal reticular apparatus. This later came to be known as the Golgi Apparatus, in honor of its discoverer. Now, let's take a closer look at the structure of the Golgi apparatus, which resembles a stack of flattened membranous sacs. There are three main parts we'll explore. Cis-Golgi network or CGN. This is the entry point of the Golgi. It receives materials from the endoplasmic reticulum for processing. Medial Golgi. Situated in the middle of the stack, this section modifies and sorts proteins and lipids it receives from the CGN. Trans-Golgi network or TGN. This is the exit point. It packages the modified molecules into vesicles for transportation to their final destinations. You might be wondering where the Golgi apparatus is located within the cell. Well, it's typically found near the nucleus, close to the endoplasmic reticulum. These two organelles often collaborate in the processing and transport of molecules within the cell. Why is the Golgi apparatus so important? Think of it as the cellular post office. It plays a crucial role in packaging, modifying, and distributing molecules like proteins and lipids to their appropriate locations within or outside the cell. Here are the key functions of the Golgi apparatus. The Golgi adds sugars, lipids, and other molecules to proteins, shaping them into their functional forms. It sorts the modified molecules and packages them into vesicles that bud off and move to various cellular destinations. The Golgi is responsible for producing secretory vesicles that contain molecules meant for export out of the cell. It also forms lysosomes, which are cellular organelles that contain enzymes to break down waste materials. Have you ever wondered how plants manage to turn sunlight into food? Picture a microscopic kitchen inside plant cells where sunlight is the main ingredient. What's the name of this kitchen, and how does it whip up a special kind of energy-rich treat? And why does most of the plant world wear a luscious shade of green? Welcome to the world of nature's solar powerhouses. Found predominantly in the cells of plant leaves, chloroplasts bask in sunlight, strategically positioned to harness the energy of the sun. The number of chloroplasts can vary, but within a single plant cell, you can find anywhere from a few to several hundred of these mini power stations. Encased within a double membrane structure, chloroplasts have an outer membrane that regulates the passage of molecules in and out, like a gatekeeper. The inner membrane is a labyrinth of complexity, forming thylakoids, stacked flattened sacs where the magic happens. These thylakoids are stacked in grana, increasing the surface area for sunlight absorption. The stroma, a fluid-filled space, envelops the thylakoids. It's in the stroma where the second phase of photosynthesis takes place. The chlorophyll and pigments within the thylakoids capture light energy in the first step of photosynthesis, the light-dependent reactions. This energy creates ATP and NADPH, which are crucial for the next stage. Within the stroma, the light-independent reactions, also known as the Calvin cycle, use ATP, NADPH, and carbon dioxide to craft glucose and other essential compounds. Chloroplasts are adorned with pigments like chlorophyll and carotenoids. Chlorophyll absorbs light, while carotenoids provide protection against excessive light and oxidative stress. The significance of chloroplasts extends beyond leaves. They generate oxygen, power the food chain, and help regulate our planet's climate by absorbing carbon dioxide. Vacuoles, the cellular compartments, come in various types, each tailored for specific tasks. In plant cells, the central vacuole takes center stage, acting as a versatile reservoir for water, nutrients, and waste. Meanwhile, in single-celled organisms, contractile vacuoles work diligently to regulate water balance and prevent cellular rupture. And in animal cells, lysosomes serve as specialized vacuoles, packed with enzymes for cellular digestion. The contractile vacuoles found in some single-celled organisms offer a masterclass in osmoregulation. By efficiently expelling excess water, these vacuoles prevent cellular overhydration. However, if these vacuoles were to malfunction, cells could swell dangerously due to water accumulation. Plant cells, known for their structural integrity, owe much of their strength to the central vacuole's role in maintaining turgor pressure. This pressure, generated by osmotic forces, keeps cells rigid and aids in supporting the plant's overall architecture. Conversely, water loss leads to drooping and wilting. Across the divide of plant and animal cells, we find striking parallels in the storage functions of vacuoles. While plant vacuoles take on the role of spacious storage units, housing water, pigments, and nutrients, animal cell storage vesicles serve to stockpile molecules for eventual release. The central vacuoles of plant cells are multitasking wonders. Not only do they harbor waste and nutrients, but they also store pigments like anthocyanins, which provide vibrant hues, and defensive compounds like alkaloids, which deter herbivores and shield the plant. In the realm of animal cells, lysosomes take on the critical role of cellular digestion. These specialized vacuoles fuse with incoming vesicles, initiating digestion and recycling processes. Similarly, plant cells harness their vacuoles for waste management and recycling. Intriguingly, not all unicellular organisms rely on contractile vacuoles for osmoregulation. Many have evolved alternative strategies, such as utilizing ion transporters or adapting to their environment's osmotic conditions. Beyond their roles, vacuoles also have a darker side. Genetic disorders like lysosomal storage diseases arise from malfunctions in vacuoles, leading to the accumulation of undigested materials within cells and causing various health issues. Drawing insights from vacuole functions could potentially revolutionize agriculture. Understanding how vacuoles manage water and nutrients might pave the way for crops with enhanced drought resistance and improved nutrient storage. Advanced imaging techniques allow scientists to witness the intricate dynamics of vacuoles in real time. Techniques like confocal microscopy and fluorescent markers enable the tracking of vacuole movements and interactions within living cells. The world of vacuoles offers surprises beyond the traditional roles. Some microorganisms utilize vacuoles for propulsion, while certain plant cells harness them for toxin storage, demonstrating versatility beyond imagination. In extreme environments, vacuoles might hold the key to adaptation for extremophiles. By regulating ions and molecules, these organisms can withstand conditions that would be fatal for others. Considering their diverse roles, it's no surprise that vacuoles have a varied evolutionary history. They likely originated from primitive compartments and gradually diversified across different branches of life. The basic responses of some microorganisms to environmental changes through vacuole activity hint at the ancient beginnings of cellular communication and signaling. Lastly, the implications of vacuoles stretch into biotechnology and medicine. Manipulating vacuole functions could open doors to innovations like targeted drug delivery, enhancing waste disposal processes, and even shaping the future of medical treatments. First stop is the unique structure of lysosomes. Picture these tiny organelles as cellular recycling centers. They house powerful hydrolytic enzymes that break down complex biomolecules into simpler components. But how does their structure contribute to this process? Lysosomes maintain an acidic environment, with a pH around 4.5 to 5, thanks to proton pumps in their membrane. This acidity activates the hydrolytic enzymes, ensuring efficient degradation of cellular waste. Now, let's explore the delicate balance between lysosomal enzymes and protective mechanisms. Lysosomes have specialized membrane proteins that safeguard the lysosomal membrane from self-digestion. Autophagy, a process where cellular components are enclosed in autophagosomes and delivered to lysosomes, also regulates their function. This keeps the recycling process in check and maintains cellular health. Moving on to the formation of lysosomes. Imagine the Golgi apparatus as a central hub for lysosomal biogenesis. Enzymes destined for lysosomes are synthesized in the endoplasmic reticulum, modified in the Golgi apparatus, and transported to lysosomes. Lysosomes then bud off from the Golgi's transnetwork, maturing into functional organelles with an acidic pH and proteolytic activity. Let's explore the strategic positioning of lysosomes within the cell. They're often located near the cell periphery and where active endocytosis and autophagy occur. Microtubules, motor proteins, and molecular motors work together to transport lysosomes along the cytoskeleton, ensuring efficient waste disposal and recycling. Time to shed light on lysosomal storage disorders. These genetic conditions result from defects in lysosomal enzymes or membrane proteins, causing the accumulation of undigested substances. Think Gaucher's disease, Tay-Sachs disease, and Niemann-Pick disease. Such disorders showcase the pivotal role lysosomes play in maintaining cellular equilibrium. Let's delve into how lysosomes collaborate with endosomes and autophagosomes. Endosomes transport materials to lysosomes for degradation, while autophagosomes encapsulate cellular components targeted for recycling. Through fusion, lysosomes merge with autophagosomes, resulting in autolysosomes where contents are broken down by enzymes. This collaboration ensures cellular renewal and waste management. This whole process is known as autophagy. It aids stress adaptation, maintains energy balance, and removes dysfunctional components. It's a key player in cellular health. Beyond waste disposal, lysosomes are versatile performers. They participate in cellular signaling, influencing processes like nutrient sensing and cell growth regulation. Lysosomes also shine in immune responses. They contribute to antigen presentation and immune cell activation, showcasing their pivotal role in defending the body. Peroxisomes, those tiny membrane-bound organelles, have a single lipid bilayer membrane setting them apart from other cellular components. This unique feature makes them distinct from organelles like mitochondria and the endoplasmic reticulum. Found in the bustling cytoplasm of eukaryotic cells, peroxisomes aren't picky about their location. They're scattered throughout, adapting to the cell's metabolic demands. The strategic distribution speaks volumes about their multitasking capabilities. Now, onto the powerhouse functions of peroxisomes. These organelles break down fatty acids into energy-packed acetyl-CoA molecules, crucial for cellular activities. Peroxisomes are the guardians against harmful compounds. The enzyme catalase within breaks down hydrogen peroxide and reactive oxygen species, shielding the cell from oxidative stress. They're not just breaking down. Peroxisomes also craft certain lipids, including plasmalogens, vital for cellular structure. Amino acids meet their fate here, being transformed into various products. Peroxisomes produce and handle hydrogen peroxide, maintaining a delicate balance to prevent cellular damage. Sometimes, things go awry. Genetic disorders like Zellweger syndrome can disrupt peroxisome function, causing a buildup of substances and leading to developmental and neurological challenges. Plant cells have their own peroxisomal story. They're pivotal in photorespiration, helping plants manage excess oxygen and convert glycolate into carbon dioxide. In response to the cell's needs, peroxisomes can multiply. This expansion addresses increased demands for detoxification or metabolic processes, ensuring the cell's well-being. Remember, peroxisomes and mitochondria are partners in crime. While mitochondria are energy powerhouses, peroxisomes tackle metabolism, detoxification, and hydrogen peroxide management. They collaborate to maintain cellular harmony. These tiny structures play a crucial role in certain organisms. Glyoxysomes are specialized organelles found in cells, primarily in plants and microorganisms. They share a similar structure with peroxisomes, which are involved in various metabolic processes. Glyoxysomes are enclosed by a single membrane and contain specific enzymes that participate in distinct metabolic pathways. Now, let's talk about where these glyoxysomes are located. In plants, glyoxysomes are most commonly found in tissues like cotyledons, especially during the germination of seeds. These cotyledons use stored lipids to provide energy for young seedlings until they can start photosynthesis. Microorganisms like certain yeasts and filamentous fungi also have glyoxysomes. These tiny powerhouses are crucial when these microorganisms grow on fatty acids as their main energy source. Alright, let's dive into the functions of glyoxysomes. The star of the show is the glyoxylate cycle. This metabolic pathway is like a recycling program for cells. It's an anaplerotic pathway, replenishing intermediates of the citric acid cycle. During the glyoxylate cycle, glyoxysomes convert acetyl-CoA from fatty acids into carbohydrates. This process is especially critical during seed germination, where stored lipids are transformed into sugars to fuel the early growth of seedlings. But wait, there's more. Glyoxysomes aren't just about building stuff up. They're also involved in breaking things down. They play a minor role in fatty acid beta-oxidation, the process that breaks down long-chain fatty acids into smaller units called acetyl-CoA. These acetyl-CoA units can then enter the glyoxylate cycle for the carbohydrate building process. The cytoskeleton is a network of protein filaments that give cells their shape and structure. The cytoskeleton is made up of three main types of filaments: microtubules, actin filaments, and intermediate filaments. Microtubules are the longest and strongest filaments, providing the cell with its shape and support. They are also involved in cell division, intracellular transport, and vesicular trafficking. Microtubules are made up of tubulin proteins, which assemble into hollow tubes. Actin filaments are shorter and thinner than microtubules, and they are involved in cell movement and division. They are also involved in forming the cell cortex, a layer of actin filaments that lies just beneath the plasma membrane. Actin filaments are made up of actin proteins, which assemble into long, double-stranded filaments. Intermediate filaments are intermediate in length and thickness between microtubules and actin filaments, and they provide the cell with strength and flexibility. They are also involved in anchoring organelles to the cytoskeleton. Intermediate filaments are made up of a variety of different proteins, depending on the cell type. The cytoskeleton is essential for many cellular functions. Here are some examples. The cytoskeleton provides the cell with its shape and helps it to move. For example, when a cell needs to move, the cytoskeleton can rearrange its actin filaments to form a structure called the pseudopod. The pseudopod then pushes against the extracellular matrix, allowing the cell to move forward. The cytoskeleton is essential for cell division. During cell division, the cytoskeleton helps to separate the chromosomes and to build the new cell walls. The cytoskeleton helps to transport organelles and vesicles around the cell. For example, the cytoskeleton can use microtubules to move vesicles from the Golgi apparatus to the plasma membrane. The cytoskeleton can also be involved in cell signaling. For example, when a cell receives a signal from another cell, the cytoskeleton can relay that signal to other parts of the cell. Cell adhesion. The cytoskeleton helps to anchor cells to each other and to the extracellular matrix. Cells are the basic building blocks of life. They are incredibly complex and versatile, and they are essential for all life on Earth. The size of cells varies greatly. The smallest cells are bacteria, which are about 1 micrometer in diameter. The largest cells are plant cells, which can be up to 100 micrometers in diameter. The number of cells in an organism also varies greatly. A single-celled organism, such as a bacterium, has only one cell. A multicellular organism, such as a human, has trillions of cells. Cells are organized into tissues, organs, and organ systems. Tissues are groups of similar cells that work together to perform a specific function. Organs are made up of several tissues that work together. Organ systems are groups of organs that work together to perform a major function of the body. Cells are constantly changing and adapting. They can grow, divide, and die. They can also change their shape and structure to adapt to their environment. Cells are incredibly resilient. They can withstand a lot of damage and still survive. However, if a cell is damaged beyond repair, it will die. Cells are fascinating and complex structures that are essential for life. They are constantly working to keep us alive and healthy. I hope you enjoyed this video. If you have any questions, please leave a comment below. And don't forget to like and subscribe to my channel, and also press the bell icon for more videos about science and biology.